Hi, I'm Roarke Horstmeyer, and this is a talk I gave at Chan Zuckerberg BioHub a couple weeks ago titled High Throughput Computational Imaging Across Multiple Spatial Scales with a Multi-Camera Array Microscope. So, what does that mean? Well, I'll explain. Basically, a lot of our research in my lab focuses on developing new types of microscopes. These microscopes that I'm showing here depict standard microscopes that really haven't changed very much over the past 200 years. So the general idea of a microscope consisting of an objective lens and then an eyepiece to look through pretty much continues on till today in many labs, where the new addition is that little tiny contraption atop the news ice microscope, a digital image sensor. So that really changes the game of what you can see and do with a microscope today. The constraints of a microscope largely are impacted by the objective lens. So objective lenses still from a couple hundred years ago until today are pretty much the same. They can see a relatively small area, a few millimeters, at high resolution, about a micrometer. And if you want to see at higher resolution, you zoom in to see a smaller area, and vice versa. And so this tiny little area that you can see generally limits the speed at which you can look at large samples, and also makes imaging live, living specimens which are moving around quite challenging because they can naturally move outside of the field of view quite easily and readily. So a lot of our research in my lab and what we'll talk about here is focuses on the design of large area, high resolution microscopes. So we're really trying to allow researchers and others in industry to see extremely large areas rapidly and efficiently, which allows them to scan across many samples quickly and allows them to complete new studies with living specimens. So why is this type of new microscope important? Well, there's a number of reasons. Here I'm highlighting 4 that we've really begun to focus on that we know it's important within because we're doing new experiments and driving new innovation in these areas. In the top left, I'm showing small model organisms like the fruit fly, zebrafish, and C. elegans. We can image those very rapidly and observe their dynamic movements and behavior over large areas and at high throughput. On the right, I'm showing a 96 well plate. So this is a standard format where you can perform high throughput experiments looking at many different samples, different perturbations across those samples. Our microscopes are very well suited to image those plates rapidly, even in a single snapshot. We also are examining applications of this new microscope to look at cytopathology slides and for microsurgery, so for surgical imaging applications. I'll focus on the first 2 topics in the remainder of this talk. So our solution to be able to see a very large area at high resolution is relatively simple. In step 1, we take the performance of a standard microscope and shrink it down into a small package on the order of a centimeter by a centimeter in size. This ability to shrink it down is quite simple once you remove the need to have an eyepiece to send the image to look at for a human, and instead solely rely on a digital image sensor. Step 2 is we put all of these little microscopes together in an array, and we call this a multi-camera array microscope, or an MCAM. Step 3 is we configure these microscopes to each image one area of the specimen plane that's unique and slightly overlaps with other neighboring cameras. They don't have to overlap, but in this configuration I'm showing they slightly overlap. We can take all here 24 image and video streams that are coming off of these 24 cameras and then stitch them together to form a large area high resolution image composite. So an image that contains 24 times more pixels than a normal microscope would, or that observes a 24 times larger area than a normal microscope. So our first generation of this MCAM technology is shown here. It contained 54 individual cameras. Each camera had 13 megapixels, so a total of approximately 700 megapixels, or 0.7 gigapixels. The field of view of this system was approximately 100 by 140 millimeters, so 10 by 14 centimeters. And the resolution we were designing towards was to look at model organisms, which is about 4.5 micrometers per pixel, or a 9 micrometer full pitch 2-point resolution. So this new technology we were able to develop had all 54 cameras fully synchronized within 6 microseconds. And when you're streaming video data off of 54 cameras, it's a lot of data. And so the total data rate of this system went up to 7 gigabytes per second. So here's an example video of just the type of area this microscope can show. I am going to preface this by saying that it's very hard to show these gigapixel-sized videos on standard screens, right, which just have a million pixels or a few million pixels, right? But essentially, a standard microscope would observe something like this, an area about 5 by 5 millimeters. An optimized microscope in this resolution might see 2 by 2 centimeters, but our MCAM can see an area shown here where we're zooming out to show the full area that we're capturing at video rate, but the resolution we obtain is equivalent to what I showed at the beginning. So in other words, just like on Google Maps, you can zoom in to any part of this whole area and see that high resolution. So we've used this new microscope for a couple interesting demonstrations. In one, we configured it to capture 3D images. And so here's an image of freely moving ants, or a video rather, of freely moving ants that we captured. And what we're able to do is slightly change the imaging plane such that more than one camera sees each point in the field of view, which allows us to use stereoscopic or photogrammetry software, which is software that takes multiple perspectives to then fuse a 3D image result. And a lot of applications of this first generation MCAM focused on imaging zebrafish larvae. So these are small model organisms that move around rapidly, and oftentimes experimentalists want to image many of these all at once. So in this video, I'm showing an example well zoomed in where we're tracking a freely moving zebrafish larvae, but we can see this across all 96 wells of this 96 well plate. So in other words, the resolution I'm showing in the middle we have across all of the wells in the background. And so we process those videos rapidly to decrease the total amount of data that is captured and has to be saved. And so here's another example where we're tracking a zebrafish larvae while it's hunting. So we can, again, perform this across 96 wells all at once, enabling new high throughput experiments, for example, to study how drugs or toxins or other interventions impact the hunting behavior of zebrafish. So some of the features we're tracking in this fish automatically include its eye angle and inner eye distance, and then its orientation, velocity, and tail movement are other features. We take about 15 morphological statistics that we're capturing across all of the wells, all of the fish in these videos, and we try to synthesize those into behavioral motifs or ethograms. We've developed software that can do this automatically. And then our software can plot these behavioral patterns to users of our microscope, where these behavioral categories offer unique insights that no other microscope could capture because, again, they either have to look at one well at a time, so performing these video-based assays is extremely tedious, or if you zoom out a lot, you don't have resolution to see these fine features that allow us to resolve these behaviors. We can also perform fluorescence imaging with this microscope. So here's an example of immunotoxicology assay performed at the Yoder lab, where a time-lapse image was acquired across a 96 well plate to look at neutrophil migration. That's what those green dots are in that inset. And again, these images we're capturing across the entire well plate to allow these more statistical significance in various high-throughput studies. So we can also configure our MCAM technology to not image a continuous area, but to instead zoom in more to see at higher resolution. So when we zoom in from where we were imaging in those previous examples at the black plane to now image at this closer red plane, you'll see that the individual fields of view of each of the microcameras do not overlap. And so this particular high-resolution configuration is really well suited for imaging multi-well plates where one wants to focus on the center of each well. We've developed two different approaches to this higher resolution imaging modality. One contains 24 cameras, and we can image all in parallel, just like the previous MCAM design. And to image a 96 well plate, we scan a little bit in a 2x2 grid. In a second configuration that's currently under development, we can pack together 96 cameras to image these 96 well plates all at once. We've developed a variety of objective lenses for these designs at high resolution, so ranging from 0.8 up to 0.5 numerical aperture, which corresponds to a 2x lens all the way up to a 20x objective lens. And the illumination becomes very important at these higher resolutions. And so we can do bright field imaging and 4-channel fluorescence imaging in these designs. Here's one example of, again, imaging zebrafish, just to have some continuity here, but to show how much the resolution has improved. And so this is a video just showing a few wells and zooming in on our interface in real time, where these 24 video streams are all being acquired synchronously, and we can see blood flow. And so on the left is the raw video data that our microscope is capturing, and on the right is a processed view to highlight the blood flow in the tail there, and then here elsewhere where you can see the beating heart, for example. So an application of this high throughput imaging technology is to look at 3D specimens. It's otherwise challenging to capture 3D data across multi-well plates. It's very time consuming. Our approach with having many microscopes packed together naturally speeds that up to be efficient and useful. So we can image 3D specimens like organoids, which are small pluripotent stem cell derived miniature 3D cell models that recapitulate the function of organs. In the middle is a brain organoid where the flashing is neuronal firing that's creating fluorescence via GCamp. In the middle, you can see movement, slight movement. That's a cardiac organoid. So that's an organoid that's actually having contractile beating. And on the right are tumor organoids that we've co-cultured with T cells to watch here in a time lapse how the T cells are going ahead and attacking those tumor cells. In all of these approaches, we do processing to take 3D data and rapidly reduce it into 2D composites to allow for more streamlined and effective assessment, again, reducing the total amount of data that we need to save in these experiments. So this technology has been productized by Ramona Optics. A new product called the Vireo now contains 24 microscopes. It can image at extremely high speeds compared to existing microscopes. It can image up to 1,000 multi-well plates a day. It can image an entire well plate with fluorescence, 96 well plate, in a minute. It can image an entire 96 well plate just with bright field capture, including Z-stacking, in about 15 seconds. So it's very fast and an interesting new technology that embodies a lot of the features I just explained. So folks are using this new technology to do screening of organoids, like I just mentioned. Here's an example of our software interface, where in this case a 384 well plate was rapidly scanned, and then our software that we've developed can segment and highlight each of those organoids in those wells. And here's another example of doing a time-lapse image acquisition of a 96 well plate, here with T cells, again co-cultured with cancer organospheres. And so in this video you'll see the ability to zoom in on any of the wells, and here we're seeing 2-channel fluorescence, red and green fluorescence, 3-channel fluorescence and bright field. And then we're developing software to be able to rapidly track and follow these cell clusters across time. So in summary, multi-aperture microscopes, I hope I was able to convince everyone, can just see more than a standard microscope. So instead of slowly scanning across a specimen one field of view at a time, looking at a little patch at each step, our parallelized microscope technology can allow you to see a much larger area or an entire multi-well plate all at once effectively. And so that speed advantage is one thing, But what that new technology really does, which we're excited about, is it opens up new assay opportunities. So now, instead of capturing an image at each location, one can capture video at each location. So with the ability to capture video of hundreds of specimens all simultaneously, we're excited that researchers can answer new questions or begin to probe new areas of investigation. And so I'd like to thank my lab for all of their help in putting this technology into useful and exciting new applications. And I'd like to thank Ramona Optics for helping productize this technology as well. And with that, I'll say thanks for paying attention. Bye.